In part 3 of this series of videos, we're going to be discussing the G protein coupled receptors pathway. Now, the G protein coupled receptors are actually the largest family of receptors in the human body, and that's because they include all of the receptors that control smell and vision. And in particular, there's a very large number of receptors that are involved in smell called the olfactory receptors. And this very large number of receptors allows us to smell a variety of different molecules. So in order to understand the G protein coupled receptor pathway, we first need to understand something about the structure of G protein coupled receptors. So if you look at this image on the right, this shows the structure of a G protein coupled receptor. These receptors are also known as 7-transmembrane receptors because they cross the membrane 7 times via 7 hydrophobic alpha helices. So basically they thread back and forth through the membrane, embedding these hydrophobic alpha helices in the membrane. However, they still have an extracellular ligand binding site on the outside that is involved in binding the ligand. In this case, it may be an odorant, something you smell. They have the transmembrane segment, actually 7 of them, and then they have an intracellular domain. And in this case, the role of the intracellular domain is to interact with a family of proteins called G proteins, hence the name G protein coupled receptors, because how they transmit the signal that the ligand has been bound is through activation of these G proteins. So let's take a look at what this looks like and how they activate a G protein. So here we have our G protein coupled receptor. Here we have our inactive G protein. Much like RAS that you learned about in the last video, G proteins are also GTPases, meaning that when they're bound to GDP, they're in their inactive state, and they can become activated by releasing this GDP and binding to GTP. So when the receptor becomes activated, it then leads to activation of the G protein by causing it to release GDP and bind GTP. Then this activated G protein can go on to activate an effector enzyme in the cell. So these effector enzymes then effect a cellular response. So we're going to talk about 2 different pathways that G protein coupled receptors can activate, but in both of these cases, this pattern will be true, that a G protein coupled receptor activates a G protein, which then activates an enzyme to lead to a response. And the signal is stopped when the G protein becomes inactivated by cleaving GTP to GDP, allowing the cycle to start again. How these 2 pathways will vary will be in the identity of the activated enzyme. So let's talk about an overview of the general G protein coupled receptor pathway first, and then we'll get into the 2 different pathways that we're going to talk about. So here we have the first messenger or the signal or the ligand. So those are all terms for the molecule that's going to bind and activate our receptor. When it binds, this leads to activation of the G protein by causing it to bind to GTP. The G protein then activates its effector enzyme, and that effector enzyme will go on to produce a second messenger. This is the major source of amplification in this pathway because, as you remember, production of a second messenger is one way that we can amplify a signal. Then this second messenger can go on to activate a kinase or some other kind of molecule that can go on to induce a cellular response. So now let's take a look at one of the specific G protein coupled receptor pathways. So this is the cyclic AMP pathway. So in this case, our major source of amplification is going to be the second messenger, cyclic AMP. The cyclic AMP pathway is the pathway used to create a response to the signaling molecule epinephrine. So some of you may have experience with epinephrine. If anyone has an anaphylactic allergy like me, you may have to carry an EpiPen, and when you inject yourself with that EpiPen, in addition to saving your life, it causes your face to flush, your heart rate goes up, your respiration goes up, and that's because it's activating this G protein coupled receptor pathway. So when epinephrine binds to the G protein coupled receptor, it activates the G protein, causing it to bind GTP. Then the G protein goes on to activate the enzyme adenylyl cyclase. Adenylyl cyclase converts ATP into the second messenger cyclic AMP, which is the major source of amplification in this pathway. Cyclic AMP then goes on to activate protein kinase A, which protein kinase A is an enzyme that will go on to phosphorylate many substrates that leads to cellular responses that vary based on what the cell type is. So for example, if it was the pacemaker cells, it would increase your heart rate in the heart. So just to review, our major source of amplification in this pathway is cyclic AMP, which is produced by the enzyme adenylyl cyclase. All right, so now let's look at the second type of G protein coupled receptor pathway that we can generate. Here the story is going to be similar. Our major source of amplification is going to be through second messengers, but we're going to use a different set of second messengers here. So this is the IP3 DAG calcium pathway. So these are also second messengers that we talked about in the first video. And one example of this pathway is the signaling molecule in this case is angiotensin. So this is a signal that helps us maintain a constant blood pressure. So this is a pathway involved in control of blood pressure. So here, the signaling molecule, like angiotensin, binds and activates the G protein coupled receptor. The G protein coupled receptor then activates the G protein by causing it to bind to GTP. This G protein then goes on to activate the enzyme phospholipase C. And phospholipase C is an enzyme that cleaves the phospholipid PIP2 into DAG and IP3. So essentially it's releasing the head group IP3. So DAG can then go on to activate other subpathways, but we're going to go on and follow IP3 because here IP3, when it's released into the cytoplasm, can bind a calcium channel in the ER and cause it to open. So this is an IP3 gated calcium channel, meaning that it has a gate that opens and closes to allow calcium out of the ER, and when IP3 binds, that gate opens, allowing calcium to flow into the cytoplasm. So then calcium can activate a number of proteins that can then lead to cellular responses like controlling blood pressure. So just to review this pathway, we have two major sources of amplification. So here we have the enzyme phospholipase C that was activated by our G protein. It produces the second messengers DAG and IP3. And then we actually have another second messenger that's another major source of amplification, and that is release of calcium from the ER through the IP3 gated calcium channel. One interesting bit about this pathway, this is actually used medicinally. So lithium is a common treatment for bipolar, and why it works is it inhibits this IP3 DAG pathway that's hyperactivated in people who have symptoms of bipolar. So now I want to take just 1 minute to remind you that you are responsible for filling out the worksheet that I have included with this set of videos. That will count as 5 points towards your next quiz if you just fill it out and fill it out completely based on these videos. And I'd also like to offer you an extra credit activity. So for 2 extra credit points, circle the major source of amplification in each pathway listed in your activity, and I'll give you 2 extra bonus points on the next quiz.